Good morning or good afternoon, everyone, and welcome to Genome Webinars. I'm Julia Caro, Managing Editor here at Genome Web, and I'll be your moderator today. The title of today's webinar is Highly Sensitive Structural Variation Detection for Medical Genetics, a Comparison to the Standard of Care. This webinar is sponsored by Bionano Genomics. Our speakers today are Dr. Alexander Heuschen, Associate Professor of Genomic Technologies and Immunogenomics at Radboud University Medical Center in the Netherlands, and Dr. Sven Bocklandt, Director of Scientific Affairs at Bionano Genomics. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which normally appears on the right side of the webinar presentation. We will ask our speakers your questions after their presentations have concluded. Also, if you look at the bottom tray of your window, you will find a series of widgets to enhance your webinar experience. With that, let me turn it over to Dr. Bocklandt for a 5-minute introduction to the Bionano technology. Please go ahead.
Okay, thank you for joining us. I'm very excited to tell you about our genome imaging technology to detect structural variation. And detecting these large structural variants in a genome has traditionally been very difficult. If a child with a genetic disease or a tumor gets analyzed genetically today, that still happens with traditional cytogenetics, using sometimes half-a-century or decades-old methods to visualize patterns on intact DNA to detect structural variation, and they're really using a microscope for that. Now, when NGS came along, there was this promise that sequencing, whole genome sequencing, would replace cytogenetics and be able to detect these SVs just as well. But turns out that they came with the wrong instrument, because instead of a microscope, they really brought a blender. And this may seem silly to you, but I would challenge you to tell me how many pieces of a kiwi are in this blender and where they are after I turn this blender on. But ironically, that's exactly what happens when you start an NGS experiment. You fragment that perfectly intact DNA into little pieces, and then you try to make sense of the large structure. And that is not the right approach. It's especially difficult because the human genome is just highly repetitive, with only about 1/3 of the genome consisting of unique sequences and 2/3 consisting of repeats. And each of these individual repeat elements already are longer than short reads, and so they can be spanned with individual reads, let alone long repeaters of that. And even long-read sequencing cannot span most long arrays of these repeat units. And that's why traditional cytogenetics still rules the detection of structural variation in a clinical setting. Now, Bionano really does the exact same thing. We visualize patterns on intact DNA molecules, but we add a nanochannel array to that. This is an array of hundreds of thousands of channels that are so narrow that single DNA molecules are stretched in these molecules and then imaged. And we image labels on these molecules, which you see in that image below. And our microscope is called Saphyr and looks like this, and it takes care of this entire process. Bionano produces this Saphyr instrument. We also have the chips that linearize the DNA. We have DNA isolation and labeling reagents, and we have our software that helps analyze and assemble the genome. Now, before Dr. Heuschen will tell you about how he uses this technology, I want to start by quickly explaining you how it works. So first, if you want to image extremely long molecules of DNA, you need to have megabase-sized molecules, and standard DNA preps don't do that. There's too much mechanical forces on the DNA, and these molecules break into fragments of 30 to 50 kb. So Bionano has released our own DNA isolation kits and protocols called Bionano Prep SP. It just takes 4 hours, needs only 650 microliters. That micro is missing there, fresh or frozen blood, or 1.5 million cultured cells, or just 10 milligrams of a cancer biopsy. FFPE samples don't work because the DNA is just too fragmented, but anything that's fresh or frozen will work great on our system. And if you compare that with a standard QIAGEN protocol that's recommended for long-read sequencing, for example, They get reads of just about 20 or 30 kb. If you look at that distribution of where the Bionano molecules fall, you see that that is on the far, far left, and that all our molecules really are larger than that. In fact, the shortest molecules that we use in our analysis are 150 kb, and then they go all the way up to 3 megabases. And our protocol is also faster than that standard protocol. So once you have isolated megabase-sized DNA, we label a specific 6 base pair sequence motif all throughout the genome. This is not done with probes. It's simply a single enzymatic reaction that attaches a fluorophore directly to the DNA at the presence of that 6 base pair sequence motif, and that occurs about every 6 kb. And then we linearize the DNA in these nanochannels. The DNA gets uniformly stretched, and we get all the information we need just by taking a picture. You can see that the images, that the molecules shown below can clearly sit in each of these channels. The backbone is stained with an intercalating dye that gives the blue signal, and that sequence motif is labeled in green. The molecules are cycled through the chips and imaged, and we can collect 100x coverage on 6 human genomes per 24 hours. We're increasing that later this year. Or you can run the flow cells longer and collect 400x coverage of 3 human genomes in much less than 2 days. Once these images are collected, we extract the digital information and do a complete de novo assembly of the genome by aligning every molecule that we image with every molecule that we image, and we build a consensus genome map shown below. And then we can compare these maps with each other or with a reference to call structural variants. All major type of structural variants are called, you can see deletions and insertions are called just by measuring the distance between these labels. We do that genome-wide starting at 500 base pairs. We can look at repeat arrays. We can detect tandem duplications, direct or inverted. We detect translocations, balanced or unbalanced, and inversions as well. And when we show you the genome in a circus plot just like that, you can simply click on any of the structural variants that you find to zoom in into the maps. Here's a translocation, and now you can even see the molecules that support that call. But all of this gets exported as a standard VCF file that you can analyze however you want. On top of that, we have a copy number tool that is able to detect copy number variants down to 10% allele fraction. And for complex cancer samples, we can look at individual single molecules and call structural variants down to just 5% allele fraction. 5% allele fraction, genome-wide, unbiased, any type of structural variant, there's no one else that can do that. Now, before we get started, I want to talk about one publication from one of our customers, Dr. Jim Broach at Penn State in Hershey, who looked at 12 leukemia genomes using our technology. And what was really interesting there is that he found variants that are not typically looked at in leukemias. For example, in this non-Hodgkins lymphoma, Bionano called an inversion in the lymphoma cells and not in the T-cell controls that broke the P10 gene, which is a known tumor suppressor gene with known compound treatments for that is never tested in leukemia. So this could really change the outcome for these patients. They also showed recurring structural variants in dozens of genes on the right that have not before been shown to be associated with leukemia, and then in tons of genes that have never been associated with cancer at all. So being able to detect these structural variants in complex cancer genomes, genome-wide and unbiased, really can lead to new biomarker discovery and potentially treatment and drugs as well. There's a webinar that Jim Broach presented here at Genome Web that is available on our website or on Genome Web, and on the Genome Web website in the white paper section, you can find a transcript from that. I want to finish by inviting you to our clinical research symposium, which happens the day before ASHG. If you point your camera at the screen right now with your cell phone, then you can register for that. But of course, you can also find this on our website. If you can't make it the day before ASHG, then join us during ASHG itself at our workshop. And again, you can register on our website or just by pointing at the screen. So have a look at our bionanogenomics.com website. And with that, I would like to introduce Dr. Alex Hoijsen, who is going to take it from here.
All right. It's a great pleasure to be invited to this webinar. Thanks to Genome Web, and thank you to Bionano Genomics and a great pleasure to speak to all of you around the globe today. So I'll speak about one of our studies where we just explored the capability of the Bionano genomic system, and I should disclose that Bionano supported us with some reagent costs. As Sven just alluded to, the classical way of looking at chromosomes was many years, actually decades, to look at karyotyping of chromosomes. Actually, I'm originally trained in the cytogenetics lab, and to me that always appeared as an art rather than a science, so it's really highly skilled people that do that. However, as Sven has just alluded, there's now novel ways of stretching really long pieces of chromosomes by the nanochannels in the Bionano system, which then can be labeled. And in essence, if you try to summarize that, what Bionano offers then is cytogenetics on steroids. And that's why I sometimes also refer to this as next generation cytogenetics, because what the output of the Bionano gives us is a cytogenetic analysis of a genome just with 500,000 bands, and overall a 10,000-fold improved sensitivity compared to classical karyotyping. And it still maintains a lot of the advantages of classical cytogenetics. So we still get a genome-wide analysis, we still can get positional information, and in the best case scenario, we can also even reach single molecule resolution. So we're really looking at individual pieces of chromosomes if you look at aberrations. I'm quite often asked why we decided to start testing Bionano right now. Although they have been in the market for several years, I think they made some important advancements, especially in the last year or so. One of which is already alluded to is that there is a novel DNA isolation method. Previously, and also for still unusual samples, sometimes that protocol is used, and that is an agarose plug method. But for our standard samples, we are now very happy and delighted to use an optimized DNA isolation that allows us to do 12 samples in a single day. And we think that is potentially even further automatable. So that is offering quite an advantage over the method that was used previously. Bionano also increased the label density in their recent release, so we now have roughly 500,000 labels genome-wide. Solution that spares genome-wide. Instrument released by Bionano now gives us the chance to run up to 6 samples a day, or if you're talking about cancer samples or leukemia samples, where we aim to detect somatic events, and those are 6 samples in less than 48 hours. One also very important aspect to us was that the pricing started to get competitive. So the current price is around $500 per sample, which is kind of comparable to standard CNV-microarrays, for example. And there's quite some room for improvement. It's interesting to see that there may be a chance to bring down the price quite significantly in the next month and years. To us, after seeing first data, we were also impressed by a few other features, and that is in particular also the analysis of samples. So to me, that made much more like a CNV-microarray-like analysis possible. In our lab, it's technicians and PhD students that do the analysis that don't have a computer background, and still they can get the samples analyzed. I think one important criterion of the higher throughput of the latest system, this Saphyr, allows very high coverage of the genome, up to 400-fold, which increases the sensitivity for somatic events very much. And to us, in our laboratory in Nijmegen in the Netherlands, the Bionano system is very complementary to the entire sequencing portfolio of short and long read sequences that we already have. From all the first data we've seen, and by a study that I will allude to in a second, our goal would be to use the Bionano to replace, by and large, the classical karyotyping fish and CNV analysis we're doing as a routine test. And to really do so, we need a proof of concept study. So we are just in the middle of this proof of concept study right now, and I'm very happy to share an update with you today. So we decided in our laboratory to test the Bionano system for 150 samples, of which 100 are leukemia samples of various different diagnoses. For all of those, usually the standard of care is karyotyping plus array plus fish, or sometimes 2 of those 3 combinations as a primary test. The standard samples we use for that are usually bone marrow aspirates or EDTA or heparinized blood. But we added 50 extra samples. Those are constitutional cytogenetic aberrations And there usually we use cell lines, either from fibroblasts or chorionic villi, or even EBV cell lines, but also fresh or frozen blood can be used for those. And those are 50 representative samples of important cytogenetic aberrations that we've seen over the last years. And again, the ultimate goal would be to implement that as part of our routine process and replace the classical methods, at least to a large degree. Here's just a first slide on the first data we received, and all the data we currently get are looking very similar to what I'm showing you here. So the average read length we can generate by this optical is 270 bases. So that is much, much larger than we used to from any sequencing. We can determine ourselves, so we can stop or run a little bit earlier. Then we generate 100x genome-wide coverage. That is usually sufficient for all germline aberrations, but we can get up to 400-fold coverage from a single chip and a single experiment. And then down below, you see all the different types of structure variants we find per sample. Here's just 4 examples shown. And usually we detect genome-wide between 5,000 and 6,000 structural variants. In this particular example, this is already 4 leukemia samples, and for those you also directly spot some translocations, which can be important drivers for leukemia. But let me show you some more details in the next slides. So first important example is a case of a CML. So by karyotyping, we already detected that there's a 3-way translocation between the current finding 2. That have worked on CML, we spot the 922 translocation probably. If we analyze DNA, high molecular weight DNA of this sample by Bionano, this is the first we will see after the analysis. So this is a surface plot, and you'll directly spot the exact same 3 translocations that were also visible in the karyotype. And just by one simple click, or from this, everybody working on CML will recognize again the 922 translocation, which is the classical Philadelphia chromosome. However, in this case, it's a more complicated 3-way translocation, but still affecting chromosomes 9 and 22 as the most likely driver. But with one simple click on the translocation line here, we can directly see what this event is. Actually, the beauty is we zoom into this, and we directly see the assembly of the aberrant molecules of the sample, and we see that this assembly line matches perfectly to the reference of chromosome 2 for the right part of the assembly, and the left part of the assembly directly maps chromosome 20. If you see the purple line in the middle, that is exactly where most likely happened, and that's the individual molecules supported this event, and by that get confidence that this is an event. Quite in this case, because we don't.
I'm sorry, your sound is cutting in and out.
Second example, right? But let's see. Do I need to?
Can you say something?
Yes, I'll try to hear.
Yeah, can you please?
This is
call in by phone.
I don't move anymore. Okay.
Can you please call in by phone? We cannot hear you.
Yes, I'll
Thank
do
you.
that. Okay.
Sorry about that.
Okay, Julia, can you hear me again?
Alex, I can hear you. Can you say something again?
Yes, this is Alex. Can you hear me now?
Yeah, the sound is great. Please proceed.
Sorry for this interruption. All right. So I'll just move quickly. So this is the second example I brought. By careful karyotyping, we can see a translocation between chromosomes 1 and 2 in this case. But usually, to reassure ourselves, we usually perform array CGH in addition. Oops, no, this arrow is not moving. We do arrays in addition, and only by that we can then spot that next to the translocation, it's very likely that we have a gain and a loss of material. The nice thing about Bionano now is that we can actually see all of that in one go. Here you see a circus plot only of the two affected chromosomes 1 and 2. The line again shows us a translocation between these two chromosomes, but if you zoom in to the CNV profile directly next to the circus plot, then we can directly see that this must be an unbalanced translocation with a net of gain of chromosome 1 and the loss of chromosome 2 material. And that was only in 50% of all cells, and 25% of all molecules. So that was reassuring that we can use Bionano for these types of samples as well. Again, also in this case, we found something new. Actually, here was a new translocation between chromosomes 5 and 14. Again, by just clicking once, we can see directly the breakpoint here, which by now was also confirmed by FISH, showing exactly the breakpoint that we anticipated from Bionano. Lastly, I brought you one X-ray example of another case where we had a complex deletion event on chromosome 20. So this was a case of a chronic myeloproliferative neoplasm. And you see the CNV array profile here with multiple deletions and multiple breakpoints on chromosome 20. What we can also doubtly read out from the array is that there's only 70% of aberrant cells, so 35% of variant allele frequency. Once we analyzed this sample on Bionano, we spotted the CNV profile, but actually what was remarkable in there was that despite several deletions being called, there were some that were visible in the CNV profile but haven't been called in the Bionano software. But luckily, Bionano just released a novel software tool that is called the Varian tool. So you see the circus plot of chromosome 20 and 3 on the left from the old software, and the newly released software that was released mid of June produced more sensitive data. And in this case, you can see more deletions on chromosome 20, but also more translocations, and I'll show you the details on the next slide. Actually, we also spotted a deletion on chromosome 3 in this case, which is highlighted right now. If we zoom into individual events, we now see that thanks to the new Varian tool, we have a higher sensitivity for especially somatic CNVs, or general structural variants. We have a much nicer visualization now of the CNV profile, which you can now see on the top. And what is actually very nice is that the CNV profile and the individual maps of the molecules down below actually support each other. So we now have many deletion-spanning molecules that support individual events. And again, here, I just show you a zoom in to a deletion on chromosome 3, which initially was not spotted by our classical methods. So this is a consequence of a translocation in this case. And importantly, this deletes a gene called FOXP1. This is not a known driver gene for leukemia, but it is known to be deleted in other cancers. So potentially this would be interesting to follow up. In other cases, we've also spotted novel events that were not visible before, sometimes simply because they were not tested for. And now with Bionano, we always get a genome-wide view instead of, for example, a limited set of FISH probes. So here, for example, we have a new fusion between BCL3 and an IGH gene in a case of CLL. Overall, we find sometimes fusions that were simply not tested for. Sometimes we find novel fusions in previously described cancer driver genes. Sometimes we also find completely novel fusions. And those are basically now visible with a one-click analysis. So click into the circus plot and directly see where those fusions are and we see how many molecules support those fusions. And I just remembered back in the old days when my colleagues were doing these kind of analysis using MFISH in combination with PCR. Sometimes it took months to just characterize one breakpoint that could potentially be interesting. And now we all see it after a simple analysis. So that's quite a remarkable progress to see all these novel fusions. All right. Now I'll switch gear a little bit and move forward from the cancer field and the leukemia fields in particular to germline structure variants, as they are also important in various diseases. Here, I again brought you two examples. So our first example is a microdeletion on chromosome 8. This is a germline event that was initially detected in our diagnostics by CNV-microarrays. If we just add standard filters on Bionano, we usually only end up with around 30 or so rare events genome-wide. And if you then click in on the event that is called on chromosome 8, we directly see support for the previously known microdeletion. So in the upper bar, you see a CNV profile, but in this case, it's also nicely supported by the assembly or the map if we compare that to the reference. So in this case, we basically get a very accurate estimate of the breakpoints by the assembly, but also the coverage-based CNV profile is supporting this event. This is basically an extra analysis you get for free, and Sven briefly alluded to that in his first presentation. I brought you another example. This is a case of classical DGeorge syndrome with a deletion on 22q11.2. Again, if we set up some standard filters, and they're usually based on frequency, then we are again just ending up with 30 events genome-wide. If you then zoom into chromosome 22, actually the CNV profile supported the previously known event. However, the assembly did not, or not fully. So we see that there's a lot going on, but to some of you it will be known that this is probably the most complicated region of the human genome. So this is surrounded by very complex repeat structures, and at least the standard reads that we currently generate don't fully bridge those repetitive regions. We are currently doing a reanalysis only using the 5% longest molecules to see whether we can identify this also in the assembly, but at least we got support by the CNV profile. Just to sum up the proof of concept study that we're currently performing, so we are only 1/3 of the way so far, but the good news is for the leukemia samples, we've identified all the previously reported clinically relevant variations as long as they had a variant allele frequency of 10% or higher. Quite often, we had to conclude that the previously called rearrangement was actually more complex after we saw it by the Bionano analysis. For the constitutional cytogenetic aberrations, only 17 samples are done so far. This included, for example, 8 clinically established microdeletion and duplication syndromes, all of which were identified back, at least by one of the two tools, either assembly or the CNV profile that I've just shown you in the previous slide. Now, having such a new technology in the lab, we also thought it would be very nice to use it to study some unsolved rare disease cases. A model disease we're using a lot in our institute is intellectual disability, or in particular, severe intellectual disability. If you're not working on that disease or may not have a particular interest in that, you may equally well consider this as a very good model disease for other severe sporadic disorders. Just to name some, like autism spectrum disorder, severe forms of epilepsy, or other neurodevelopmental disorders. A cohort we've studied over many years now consists of roughly 1,500 patients with severe intellectual disability. And whenever a new laboratory test came available, we tested this cohort or a representative fraction with the latest tools. So, for example, in the I would say in the dark ages of human genetics, usually clinicians asked for a single gene test. By and large, those were not very successful, except for a few genes. But usually, in such a large cohort, it only delivers a diagnostic result in 1 to 5% of cases. Then, in the mid-2000s, genomic microarrays for CNV profiling came up, and they usually solve anything between 10 and 15% of your cases. Then, exome sequencing was established also in our routine lab since 2012. And depending on your previous tests that are already done in your cohort, you can find between 20 and 50% of cases or find a mutation that can explain the disease in those cases. Then in 2014, we did the first pilot of patients that were negative for all previous tests by whole genome sequencing and solved additional patients. But we certainly didn't solve all of them, because if you now would do the latest tests that we currently have up and running in the laboratory and just do our utmost best to analyze all patients, the maximum yields we could get for such a cohort would be 62%. It is remarkable that almost all the mutations that we consider disease-causing right now are actually de novo mutations. So several of those are de novo point mutations, but also a substantial fraction are de novo structural variants. However, and sad enough, there is roughly 40% of cases where we can't give a diagnosis even after genome sequencing nowadays. So the big question for that is, what have we missed so far? Have we actually fully analyzed the genome? And there's multiple reasons why we haven't found a diagnosis in those cases. Maybe our interpretation is not good enough yet. Or maybe, and as geneticists we may not forget that, maybe some of our patients simply don't have a genetic cause of the disease at all. But I think one likely idea or one likely explanation, what we may have missed, is actually more of a technical thing. Maybe we simply haven't sequenced the mutation itself. Or, maybe equally likely, there was a structural variant that remained undetected with the current genome sequencing or exome sequencing. So that was one of our working hypotheses. So could there be any hidden de novo structural variants in the unsolved ID trios after genome sequencing that may explain disease? So that was a small study we started, and initially we thought we'll go for long-read genome sequencing. So we used the PacBio system and did smart sequencing on 5 patient parent trios in which the disease cause in the patient was not found yet. Those were done with 15 to 40x coverage and on average 13 kb reads. So really long-read genome sequencing. We found a lot of novel things by doing long-read sequencing in those samples. And here's just the highlight of our findings. So we find roughly 25,000 structural variants that are 50 bases or larger per genome. In addition, we even find up to 40,000 indels that are between 20 and 50 base pairs in size. If we compare that to standard Illumina sequencing, usually 60% or more of those events have not been called initially by the short-read genomes. However, to our own disappointment, we were not able to find a single de novo structure variant in any of those 5 patient parent trios. And we also did not find a likely pathogenic mutation in any of those cases so far. What we've done now is actually analyze the exact same 5 trios with a Bionano system. Here you just see an example of a 32 kb deletion, which is called by PacBio very convincingly in the upper panel, and the lower panel is showing us the very same deletion in a Bionano call. And there you see the assembly again, supporting that event. Overall, if you look at all the large deletions, that's easiest to us to compare, then we see very high concordance between the long-read sequencing and the optical mapping by Bionano. And actually, that echoes very much a recent publication of the Human Genome Structural Variant Consortium. And there's a paper published by Mike Chasen and his co-authors, at that time in Evan Eichler's lab. And they actually also show that optical mapping shows extremely good concordance for large structural variants compared to sequencing. I should, however, point out that the Bionano data used in this paper were from the old generation, so probably the latest Saphyr with the latest chemistry may increase the sensitivity even further. So that is actually very nice to see. Now coming back to our samples, I'll show you just one example for one of the 5 patient parent trios that we've analyzed. So here you see the results of one of those samples. So again, genome-wide, if you look at all the structural variants, we usually find 6,000 or so structural variants by just one very simple filter, and that would be that the variant was never seen in any of 200 controlled genomes that are provided by Bionano. Then we actually just end up with anything between 30 and 50 structure variants genome-wide. But then we've also done the parents, and there is a trio analysis mode in the software included. So if you then subtract all the structural variants that are seen in either of the parents, in this case, we're only left with one candidate de novo structural variant. And I'll show you that one on the next slide. Actually, this one ended up in the most complicated region of the human genome. This is just downstream of the colon de Vries microdeletion syndrome region, 17q21.31. Here's a screenshot of that. This is an event originally called as a deletion, affecting a gene that is known as NSF. If I just show you the next slide, this is the PacBio data, and actually we see that there's very low coverage in this very same region. One reason may be that the PacBio data has been mapped back to the wrong reference. So there is alternative alleles available that have been published by Evan Eichler in this lab. So we also tried to look into that, but we still don't have support for that event in the PacBio reads. But that is not very surprising, seeing the complexity of this region of the genome. If we then look in greater detail into the call of Bionano, then there's two options for how this event looked like. It could be one or two breakpoints, actually upper and lower panels shown here. But in either of those options, it is most likely that a partial duplication of these locus is disrupting actually both NSF copies on this allele. We also don't see any support in the parental assemblies for this event, so it makes it very likely that this is a true de novo event. Whether it's related to disease is very difficult to judge at this moment. It's important to know that NSF actually shows multiple copies already in the general population, so it's a copy number variable gene. But to my understanding so far, there was never an individual reported with just one allele, which this individual may carry. So that's certainly very interesting to follow up further. Then I want to close with one extra example. Actually, I got in contact with my former colleague, Roland Kuiper, who's now working at the Prinses Maxima Center in Utrecht, also in the Netherlands. And Roland has some interest in a very severe, very rare disease called atypical keratoid rhabdoid tumors. This is a very rare and aggressive form of tumors. It's usually diagnosed in very early childhood and is usually a brain or CNS tumor that is presented in the child. And the cause of that is usually very well understood. This can be caused by mutations in a classical tumor suppressor gene, either by germline or somatic mutations in this gene called SMARCB1, which is part of the SWI/SNF chromatin remodeling complex. And Ronald has seen a family in which a 1-year-old girl died, unfortunately, of this very severe condition. But they had a strong suspicion for a genetic predisposition. The parents had another wish for another child, and analyzing the tumor actually has shown that there was loss of the SMARCB1 protein by immunohistochemistry. But the question remains whether this is a pure somatic event in the cancer or whether there is already one allele affected in germline, which may offer a great risk for recurrence. Roland and his team have already studied this case extensively over the last years and months, and actually Sanger sequencing, MLPA, and even whole exome sequencing that was performed diagnostically did not deliver a germline mutation in this case. They continued to even do genome sequencing, but still couldn't find a germline mutation in this particular case. We then performed optical mapping of an EBV cell line for this case, received more than 300x genome coverage, and just did the same filtering for rare structure variants genome-wide, which in this case delivered 35 events, 35 rare structure variants genome-wide. If you then zoom into the SMARCB1 locus, what we directly spotted there is actually an insertion of 2.8 kilobases. Here with the two arrows, I'll highlight the wild-type marker distance in the reference, which should be close to 5 kb. And if you look at the assembly and the individual molecules of this sample, then we see that there's 2.8 kb inserted, including one extra label. So this insertion, we don't know where this material comes from yet, and validations for that are actually at the moment ongoing. But this insertion happened between intron 1 and 3. That's how accurate the breakpoints are at the moment. So to our opinion, this very highly likely could be the germline mutation that gives a very strong predisposition to ATRT. When we shown these results to Roland and his team, he also told us that actually they already did cDNA sequencing and saw possible skipping out of exon 2 in this patient. So in the lower panel of the patient, there is an extra banding pattern in just a small percentage of the material, which may support this finding, actually. So we believe that very highly likely we found the germline SMARCB1 mutation in this case that gives a strong predisposition. And it's important to tell you that this was really missed by all the previous approaches, and that included exome and genome sequencing. With that, I'd like to wrap up, or actually I wanted to use this last case of Roland to just highlight that we're currently launching another research program where we think optical mapping can lead to other scientific discoveries. So those are, for example, cases or families with a known linkage region for their disease. Sometimes that is already known for years, and none of the sequencing approaches have yet identified the disease-causing mutations. We're also using optical mapping now for some research families in which we suspect very strongly recessive disease. So far, we've identified a pathogenic mutation on one of the alleles, but the second mutation remains undiscovered, and we think Bionano may discover those. And then we've also launched some other research projects for samples collected by our own clinicians and researchers, but we're also involved in larger consortia like the Solve4D consortium and the Chromosomics initiatives, and we're looking whether optical mapping is also a valuable tool in these consortia. That brings me really down to my summary and my outlook. I hope I've shown you some of the excitement that we've seen with optical mapping. In my opinion, optical mapping may truly allow a cytogenetics revolution in the next years and really allow what you could call next generation cytogenetics. We think that the first data we've seen encourage us to believe that this may replace a lot of the classical cytogenetics technologies in the next years. So far, we've identified back all the previously identified clinically relevant structural variants in the leukemia cases once they have an allele frequency of 10% or higher. Especially the latest add-on on the increased throughput of the Saphyr system that allows 400x coverage allows us an improved sensitivity for somatic structural variants. And we look forward to even higher throughput that should even improve this further. And next to the improved throughput, also the latest released rare variant tool gives us very promising results, especially for somatic structural variants. And I think I've just shown you two examples today that encourage us to use optical mapping to discover structural variants in some of our unsolved research samples. With that, I come to the acknowledgement. I'd like to acknowledge Roland Kuiper and his entire team from Utrecht, and an entire team of people that got involved in establishing the Bionano in our lab, and many colleagues that have supported us over the years. And I'd also like to thank Bionano for engaging in our collaboration. With that, I'll give the floor back to Julia, and I'm very happy to answer some of the questions if possible. Thank you very much.
Thank you, Alex, for this nice presentation. As a quick reminder, if you have a question, please type it into the Q&A panel. And also, we would like to ask all of you to take a brief moment after the webinar has ended to take our exit survey and provide us with your feedback. So with that, let's get into the Q&A. So we have a couple of technical questions that I guess Sven might be the best equipped to answer. One is, will buccal swabs work as a source of DNA? And also, what is the minimum amount of DNA you need to put in?
Good questions. We have worked with mouthwash samples in-house. We haven't actually tried buccal swabs, and we don't have any commercially available protocols for it. However, we don't see a reason why it wouldn't work. It's just some work that we still need to do. About the amount of input DNA, we load 80 nanograms of DNA into a flow cell, and that can collect up to 400x of coverage from that amount. Now, keep in mind that DNA that you already have isolated and is in your freezer is not going to work. It's just too fragmented. So that's why we're stating input requirements of samples and tissue, 650 microliters of blood, 1.5 million cells or 10 milligrams of tissue. That is a large excess, so there's a lot of redundancy built in there to go into the labeling reaction and then eventually go into the chip. But so 80 nanograms is what we load.
Thank you. Question for Alex. You talked about the studies you're currently conducting, leukemia and germline samples, and that you haven't completed it yet, only a fraction of the samples you want to analyze. Is that because you're still collecting those samples, or is the reason that the Bionano system is slow?
So actually, the other samples are ongoing at the moment. So actually, we only got the system in March, and we got the entire team trained, which actually went very smoothly. And all the other things are ongoing right now. So actually, for the leukemia, a lot of the samples we run in parallel. So the classical routine tests are still ongoing for some of them, and then remaining material is put onto the Bionano. So the overall goal is to finish this entire study by the end of the year to really draw major conclusions how we move forward.
Find that. And a related question, in your constitutional samples, are there any known balanced translocation carriers?
Yes, we include some translocation carriers in there. I don't have the entire list with me at the moment. Those have been selected by my colleagues from routine diagnostics. But I think one thing that is important to mention is that translocations, the fact that centromere or very near to the centromere may not be detected. So we are already aware that, for example, Robertsonian translocations cannot be detected. And that is something that also Bionano declared themselves. There is some hope for the future if somebody comes up with smart extra labels in the centromere. I think there may be even options to discover those. But in the current setup, we would be unable to find so-called Robertsonian translocations, or assuming also other translocations that affect the centromere.
Okay, thank you. Question for Sven. What's the minimum coverage needed to make accurate structural variant codes in oncology samples? And also, can the technology be used to detect cell-free DNA, or circulating tumor DNA?
Yeah, so for complex cancer samples, we're recommending 400x coverage. So that is a single flow cell at a cost of $500 US. If you really want to find variants that are present at lower allele fraction than the 5%, you can simply combine some of these flow cells. It would double the cost, but also you can go down to 2 or so percent variant allele fraction quite easily. Sorry, what was the other question?
About the use in
cell-free
DNA or circulating tumor DNA.
That's right. So cell-free DNA is really highly fragmented. It's typically less than a few hundred base pairs. So there's really nothing that we can do with that.
Okay, thank you. Alex, question for you about the concordance for structure variants between PacBio and Bionano. So you said it was about 90%. Is there any particular class of SVs that didn't have good concordance?
So to be honest, we mainly focused on the deletions because they're easiest to compare. And some of the data is extremely fresh. So that was the main focus. Comparing insertions is certainly a bit more tricky in the sense that we don't necessarily know what is inserted in Bionano, depending on the size, but small events, we don't necessarily know where the material comes from. But the paper by Marchesen and colleagues pointed out that sometimes Bionano may even have higher sensitivity than any of the sequencing platforms, especially for insertions. And I think the reason may be quite logical. They are kind of independent of the sequence context. But I personally, on our data, we haven't done that concordance. However, we see that the Mendelian inheritance for the vast majority of events that are called by Bionano are actually in absolute concordance. Also clearly seen by our trio filtering. So usually we only end up with 0 or 1 or so candidates de novo. So that already makes us quite confident that the vast majority of Bionano calls are actually true.
Question for Sven related to the DNA sample prep. Actually, a couple of questions. One is, does your DNA prep work for fresh frozen buffy coats? And the other one is, does clotting of the blood complicate the extraction protocol, and can bone marrow be used?
Buffy coats are very straightforward. It's actually really an ideal input tissue for us. We can even work with frozen whole blood, which is more complicated because the red blood cells are in there. But buffy coats work great. Clotting of the blood. So I'm not particularly sure. We can work with EDTA and heparin preserved blood samples. We do work with bone marrow aspirates all the time. We actually have protocols optimized just for that.
I may add to that. I think we've tested both sample types, fresh and already stored EDTA and heparinase blood. And I think we even deviated slightly from the Bionano protocol by sometimes adding a DNA stabilizer even after thawing the sample. And we had quite good results with that as well. Although I think it's not fully supported yet by Bionano's protocols, we still saw good results with those sample types.
Thank you. Couple of specific questions for Alex about the two of the cases you presented. One is, how large was the de novo structure variant that you identified at the 17q2131 region?
I don't know by heart is the honest answer. It actually was called as a deletion. But actually, by looking into greater detail, it most likely is a partial duplication only of an event, which netto results in disruption of both copies. And that fragment that is first partially duplicated is tens of kb, so it's relatively large. That is the answer, but I don't know by the top of my head how large exactly.
Okay, that's fine. The other question relates to the ATRT case. The question is whether the whole genome sequencing for that was done with PacBio long reads or with short reads.
That was Illumina short reads, so more classical NGS approach, yeah.
Wanted to clarify. There's a question about, not entirely sure, I get this, but I read it to you. So it seems there's a curation burden with fewer resources than the SNP and indel type NGS databases. How are you addressing that if there is such a curation burden? And also, how is the nomenclature being standardized for this type of technology?
Ooh, that's not necessarily an easy one, and I'm not fully sure whether I understood it correctly. But yes, I think databases at the moment are heavily biased for point mutations. Obviously, there we have greatest experience as a field, and I think they're pretty accurate. There is certainly also various types of databases already for structural variants, but again, those may be biased by the so far mainly used technologies, and a lot of that is short-read genome sequencing data. So I wouldn't be surprised indeed if novel technologies that have other biases or no biases would identify novel structure variants that are completely underrepresented in databases. But what we see, if we use the Bionano, there's 200 controlled data sets already made available by Bionano, and that already filters down to 30 to 50 events in a germline case. So that seems to tell me that we already reach a number of variants that we can handle per case. So 30 events per sample is not too much to start an interpretation upon. I would, however, expect if we increase the number of genomes, especially population-specific genomes, then probably the number of rare events per genome is further lowered, and that would make interpretation even easier. I think the example of the trio has shown that de novo structure variants seem to be extremely rare. So for some research sample where you expect a de novo occurrence of the disease and de novo occurrence of the mutation, it may not be a too bad idea to actually run a trio, because you directly see that we filter down to 0 or just 1 event in a trio approach, which to me makes a very good impression in terms of how good the qualities of the system, how much we can filter out in the parental genomes to really make interpretation of the structure variants much easier.
Another couple of rather technical questions for Sven. One is, can you determine telomere lengths with this technology? And also, what is the resolution in kilobases of the technology? You might have mentioned that before.
Yeah, for telomeres, there's actually quite a few of our customers who are using our technology to look at least subtelomeric repeats. So if you go to our website in the resources section and look at the publications and just search for telomeres, you'll see some of those. And one method that one of our customers used was by actually custom labeling these repeats in or near the telomeres with a CRISPR-Cas9 based labeling system. That's not commercially supported by us, but they did that in an incredible way to look at the telomeres. The optical resolution, so when we measure the distance between two labels on our system, we can do that to an accuracy of about 50 base pairs in our consensus genome maps. Now, we call structural variants automatically starting at 500 base pairs for insertions and deletions. And we have some breakpoint uncertainty. So we can place these structural variations with a breakpoint uncertainty of about 3 kb. I think that's why Alex Hoygen said that it's like cytogenetics, but 1,000 of times higher resolution.
Question for Alex. What, by and large, do you find most compelling about the Bionano technology? Is it the ease of use for cytogenetics, potentially in the future routine use, or is it the fact that you can find rare structural variants that could unlock new understandings of disease?
So scientifically, obviously, the latter. So that is always very exciting, even in cases where we've studied a lot. If we now finally find the disease-causing mutation, that is, very rewarding to us, and ultimately also very important to the patients and families. So personally, I got most encouraged by these kind of findings, and hopefully we'll get many more in the next weeks and months. However, also from the lab side, or the way we established the system now, I was impressed by the ease of analysis. So while for other sequencing-based technologies, we really needed a great team of bioinformatics support, which we luckily have in our hospital. But for the Bionano, usually it's the people that do the lab work also start all the analysis and look into the data immediately. So to me, that is probably at this moment one of the most exciting advancements that Bionano has made over the last year or so, to make this analysis easy enough that lab personnel that previously did cytogenetics analysis or array analysis can directly work with the system.
Related question, how long did it actually take your lab to get up and running on the Bionano?
So there the honest answer is right in the training run, we directly were better than specs initially. So directly the first run succeeded. Since then, I think we had one small batch of chips that was defective, just to be very open here, that did not result in fantastic results, but that was solved and replaced immediately. And I think we had, at the beginning, we played a little bit with the sample amount input, and we went lower than what Bionano recommended. And there we had 1 or 2 samples where we didn't get enough DNA or successfully labeled DNA out. But for all the other samples, we succeeded, and yeah, that happened directly after training. So that was very nice to us.
Okay, thanks. One last question for Sven. Have you compared structural variant calls that were made by Bionano and Oxford Nanopore, where you use the Bionano extraction method, or are you planning to do such a study in the future?
We haven't done these comparisons directly, mostly because from what we've seen, doing genome-wide automatic calling of SVs with nanopore data just doesn't work yet. I mean, the most recent paper that I've seen from a group at the University of Antwerp said that they saw thousands of false positives and thousands of missed calls. So I think until these algorithms get better, I think it's going to be very hard to make these comparisons.
Okay, thank you. That's actually all the time we have for questions today. So let me thank our speakers, Alex Hoygen and Sven Bockland, and our sponsor, Bionano Genomics. And if we didn't have time to get to your question today, we'll try to have one of them follow up with you. As a reminder, please look out for the survey after you log out to provide us with your feedback. And if you missed any part of this webinar or you would like to listen to it again, A link to an archived version will be emailed to all participants today. So with that, thank you very much again for attending this Genome webinar.